So it's my incredible privilege to introduce our speaker today, Professor Naomi Zimmerman. I love saying that. I think we're incredibly proud of Naomi, who did her PhD with us, then went down and did a postdoc at Carnegie Mellon and went on to incredible things at UBC. And she's agreed to grace us with some of her time and some of her wisdom to come back to U of T and give us an update on what she has been working on in between all the various crises that we know have been going on in BC, which really has had quite the record year for climate-related incidents. So Naomi's going to talk about air quality and health, odor, and equity in terms of cannabis cultivation. So we're super excited to hear about that, but also super excited just to have Naomi virtually back with us for an hour. So with that, join me in welcoming Professor Naomi Zimmerman.
Oh, thank you. Thank you so much for that kind introduction, Greg. I think it was overly effusive, but that's okay. Also, I was like, oh yeah, having me back at U of T. I would love to actually be back in Toronto, because normally these speaker dinners in the olden days would involve, having a nice beverage and stuff after the talk. And one of these days we will do that again, but that's okay. Also pleased to see some of people attending from Metro Vancouver, which is great because I can give them some project updates at the same time while we do this seminar, which is very convenient. All right, so hi everybody. Thanks so much for having me at the Socaar seminar. I am really delighted to be able to join today and share some preliminary results from what is an ongoing project. So you'll see, a lot of this is very open-ended, and that's quite intentional because there's just so many opportunities. So if this is a question you're interested in looking at, that's great. And also, feel free to reach out because, we just started this thing as a very small scale, and we would love to expand going forward. All right, and so while I am giving this talk, I certainly don't want to give anyone the impression that this is all me. We actually have quite a large collaborative team. So this is the team minus me. It's led by an investigator team of Amanda Jang in the Institute for Resources, Environment, and Sustainability at UBC and myself. We also have Sarah Henderson, who is the Scientific Director of Environmental Health Services at the BC CDC and is also an Associate Professor in the School of Population and Public Health. We have a fantastic collaborator, Angela Eichelbosch, who is an Environmental Health Knowledge Translation Scientist at the National Collaborating Center for Environmental Health. And then really just like a fabulous team of undergrads and PhD students and postdocs who have worked on all of these things. So I just want to highlight that, while I am showing this, this is really like a team effort in the truest sense. This has been one of the nicest and most amazing teams of people that I have had the privilege of working with at UBC, like really earnestly, like fantastic group of people to work with. So before we start our discussion on, emissions and the cannabis cultivation sector, I just wanted to give a little bit of context on what we do in my lab group. So, a good lab group needs a good acronym, and I don't know if I can ever find something as good as Socaar, but I run a research group that I've called IREACH, which is the Interdisciplinary Research in Energy, Air, Climate, and Health. I wanted something that would cover a whole career, so it has lots of really big pillars of words in it. The iReach lab conducts research on the development and application of real-world based tools to quickly and ideally quantitatively assess the impact of our policy and technology decisions on air pollution and climate outcomes. We do very limited lab-based studies. We primarily use the natural environment as our laboratory to study emission sources and their impacts under changing circumstances. We're also really motivated by knowledge translation and making scientific findings accessible to policymakers and stakeholders, which is why we're really excited for this project to have like someone who does KT for a living, like part of the team from the get-go. So I think it's really helped guide us in terms of thinking about how we look at the data. So the outline for today's talk, we're going to touch on 5 different things, hopefully. So I'm going to give you a little bit of context for cannabis cultivation, some trends and importance. I'll talk to you a little bit about a broader project that we're using to sort of look at the cannabis sector, which is the Smell Vancouver project. I hear someone maybe not muted, so maybe we could just check that. Going forward, we will do the screening methodology for odor source identification for cannabis facilities specifically. I'll talk a little bit about a critical review that we just finished of cannabis cultivation emissions and some identified fronts of action. And really, this is, again, a call to researchers who are interested in this sector. Lots of different fronts of action identified. And then I'll talk about some research on the horizon. Ideally, we would have started some of this research, but obviously we're all, operating in a pandemic regime. So a lot of these things that we'd hoped to start last year, are going to hopefully be happening in 2022. So let's start with cannabis cultivation in Canada and some trends and importance. So in 2018, as many of you likely know, cannabis was legalized at the federal level in Canada for medicinal and recreational purposes. It's like one of, I believe, only 2 countries in the world that had legalized this at both medicinal and recreational levels at the time. And there are almost 600 licenses for cannabis cultivation in Canada, and roughly a quarter of these licenses are in BC. So we can see here that sort of over time, the number of federal license holders, and you can sort of see this exponential growth after 2018. And the key thing I want to emphasize is this is a fairly new legal sector, I should say, and there comes with that substantial knowledge and policy gaps. So this growing industry, a few things popped out to us when we were developing this grant and research project that we've been working on. And really, it's that this growing industry is resulting in increased odor complaints and potentially increases in ground level ozone, although we don't really know the extent of what that might be. So if you have ever been in the presence of a cannabis plant, you will note that it has quite a distinct odor. And Metro Vancouver reported, I think, over 300 odor complaints in 1 year related to this sector. And of those odorous things, we have good evidence to believe that many of these are volatile organic compounds, which we know are related to the production of things like ground level ozone. And if we look at a map of Metro Vancouver, here we have, this is not an extensive list, but we had sort of an undergrad doing some sleuthing around, some known locations of cannabis cultivation facilities. We can see that a number of them in the Metro Vancouver region are in a VOC limited region. So a region is called VOC limited if the emissions of VOCs are low relative to that of NOx, which something is released in a combustion process by automobiles, for example. And in VOC limited regions, secondary pollutants such as ground level ozone can be produced by even small increases in VOC emissions. So by having more of these cannabis cultivation facilities present, we expect there could be increased VOC loading in the region, which might have implications for ozone. It actually can be a little bit more complicated than that. In addition to just this ozone effect that we anticipate, we can also potentially have other things, like if we have ozone and sunlight and all these VOCs, there is complex nonlinear photochemistry that can be at play. We know that VOCs are important precursors to secondary organic aerosol formation, which can affect overall PM 2.5 concentrations. However, the extent of this is essentially completely unknown. And we know that many of these emitted BVOCs, as I mentioned, are odorous and can be linked to potential spike in odor complaints. Again, our knowledge surrounding the magnitude of these effects is extremely limited, which is why our research team is starting to look at some of these questions, although again, it's a huge area and we could use lots of collaborators and other people working on this sort of thing. I also want to mention important context for odor specifically. So this was something that was new to me, or a new area for me to learn about. But one reason we're also interested in odor is that it is generally treated as a nuisance. So, the population's most common experience of what they might consider to be poor air quality could be something that they experience as, smelling poorly. So odor, people's relationship to air quality and odor are intricately related, right? Odor complaints are one of the most common kind of air quality complaints from the public. However, this issue is typically treated as a nuisance. And regulatory frameworks typically focus on annoyance versus health risk. So we're trying to better measure air pollutants that are associated with odors to try to help understand or decouple some of this link between, annoyance versus specific health risk. And in some places, odor impacts have disproportionately impacted low-income, racialized, or other marginalized communities, which can make this an environmental justice issue as well. So hopefully that gives you good context for the motivation for our research, which is that empirical data on the air quality of cannabis cultivation are virtually non-existent. Further clarity is needed on whether BVOCs emitted during cannabis cultivation will increase harmful ground level ozone and particulate matter. We know that odor complaints have been increasing and the health impacts of these odors are not well understood. And I think the key thing here is that the longer that we go with incomplete policy frameworks, the higher the risk that systems will become entrenched and difficult to change. So the best time to be implementing, good policy is now, before we've sort of set precedent on what policies are going to be, and then they can be sort of more stagnant or difficult to adapt. Okay, so with that context in mind, I'm going to introduce you a little bit into the Smell Vancouver project. So this work is all funded by a New Frontiers in Research Fund grant, which requires research projects that cross traditional boundaries between NSERC, SSHRC, and CIHR funded work. So here we're really focusing on transdisciplinary mixed methods approach to understand links between odor, air quality, and community health. So what this means in this case is sort of broken it down by what I would call, let's see if I can get my laser pointer here. There we go. We're calling this more in the SSHRC domain of working on building community engagement apps that people can talk about odor and their experiences of odor. Especially here, we're focusing on the cannabis cultivation facility, but we are actually collecting data from all sorts of sectors in addition to cannabis cultivation, like sewage, for example, which comes up a lot in the Metro Vancouver region. In our sort of traditional NSERC domain, which is really my area of maximum comfort, is working on developing new techniques for measuring emissions, measuring air quality around facilities, and working with Amanda Jang to do computer modeling to predict regional impacts and assess effectiveness of abatement tools. And then lastly, from a health perspective, and really this is being led by the BC CDC aspect of the team, is thinking about things like mapping zones of impact, evaluating health susceptibility of populations living in impacted areas, and again, engaging with the community to understand impacts of this industry and other odorous industries on perceived health and well-being. So we're actually getting some of this self-reported health data from the mobile app as well. So the Smell Vancouver or Smell Van mobile app was developed in the summer of 2020 by a very talented computer science undergraduate student named Andre. It launched on December 8th in 2020. It's based on existing, or very similar to, we didn't either create the idea of a smell reporting app. There are other examples out there. So if you want to build your own, there's actually from I think the Smell Pittsburgh app, some existing code that you can start to leverage to build your own. So if you want to build one, it's definitely possible. And we're also using it as part of a broader assessment on links between odor, air quality, and health. But I'm going to talk specifically today about supporting our assessment of odor impacts of cannabis cultivation. And if you want to check out the Smell Van app, you can see it at smellvancouver.ca, and you can see where odor reports have occurred. Some statistics on Smell Vancouver. This hasn't been updated since October, and we actually had a big push in December where we had a bunch more reports just because we had a CBC article and then all of a sudden there was a whole spike of reports. But we've had over 600 odor logs to date as of the end of October. Our highest level of engagement was when we launched in December 2020, and we also had increased use in the summertime. In the summer, we weren't actually really doing any media around this, so we actually think treat this as more of a real spike in air pollutant measurements. And as we analyze this, we are tracking any sort of media or mention on social media to help us sort of adjust links between number of odor logs and other sort of air quality events. So I will say it is opportunistic sampling. The number of reports does not necessarily reflect increased odor events, but we are conducting some correlation analyses, for example, to weather currently. And there was an AGU poster about this that happened in December. So if you attended AGU and you want to see it, The paper number is there if you want to jot it down, or you can email me and I can send you a copy of the poster where it was looking at sort of all of these odor reports and not just cannabis specific ones. All right, so that's I've given you some context for what the SmellVan app does. And now I'm going to talk to you a little bit about how we can actually use some of this odor reporting data and link it to the cannabis cultivation sector. So here I'm going to be talking about a screening methodology for odor source identification from the cannabis cultivation facilities. And this work was led again by a very talented undergraduate student named Rochelle, and is being continued by a postdoctoral fellow named Sahil. Okay, so within the Smell Vancouver app, people have the opportunity to sort of log the characteristics of the odor. We have some predefined options, and then people can also do some free text entry. But we are generally associating skunky odors with cannabis. So you can sort of see a map of where these skunky odors are getting reported. And approximately 10% of all of our odor reports are being classified as skunky. One thing I do want to point out is that we do have a high density of skunky reports in the Delta area in South Metro Vancouver. And if I bring you back to this previous map, I had shown you about some identified locations of cannabis cultivation facilities. We do have a cluster of them in this Delta region. So very similar cluster between the cannabis cultivation facilities and some of these skunky reports. And that's all well and good. We can sort of see maybe there's some visual correlation there, but we want to understand if we can go a little bit further into this. So this was done, this work was really more so led by Amanda Jang, who has a lot of modeling expertise, but I will do my best to sort of summarize some of the work that went into this. But what was done was to try to more quantitatively link odor reports to cannabis cultivation facilities. Some high split modeling was done. So here the inputs to the high split models are in purple. So we have meteorological data and then our odor reports, which provide the receptor location and time. And then we also have identified some CCF locations that are also input into the data. So what high split has done is to create these back trajectories, and then we can also look at the CCF locations and compare if there are intersections between these back trajectories that we're generating and these CCF locations. We're not using a single back trajectory. We're going to use a meshed grid of back trajectories to do this, and I'll just walk you through a little bit this method in some detail. So for a little bit of context on high split, for those who might not be familiar, it is a commonly used transport model in atmospheric science. It uses Lagrangian methodology for back trajectories, which essentially is a moving frame of reference to calculate trajectories due to advection and diffusion of air parcels. And it does also consider weather by blending observed radar data with past weather forecasts. The 3D calculation that's done by high split is composed of two steps. The first step is an advection step, which is essentially the trajectory calculation component using the mean wind field from the input meteorological data file. And in the second step, this is adjusted by turbulent wind. So here, X and Z correspond to the horizontal and vertical positions respectively. And our U prime and W prime here are the corresponding turbulent velocity components, which are calculated based on meteorological data. The key thing here is that for backward in time modeling, which includes dispersion, the dispersion equations are applied to the upwind trajectory calculation, assuming that the equations are reversible when integrated from T to delta T. However, the mean trajectory should not intersect with the ground level, at which point the calculation becomes irreversible. So that is a key consideration, that the trajectory can't hit the ground. Otherwise, this assumption of reversibility will fail, which becomes important for some of our assumptions. Here. So as part of what this creates is a bit of a compromise in terms of the height of the receptor. So if we put the receptor, the physical situation, the height at which the user smells the odor is going to be a little bit closer to the ground. But but we know we have these model constraints that we need to prevent the trajectories from hitting the ground. So our compromise here is that we've run high split for 2 receptor heights above ground, 10 meters and 25 meters, which we know is not exactly representative of where the receptor is when they're smelling the odor, but we're doing this to try to minimize, having all of our back trajectories hit the ground and then the math kind of collapsing. It's also done with a meteorological ensemble. So the weather data here is the archived high-resolution rapid refresh model, which is a 3-kilometer, 1-hour resolution. And as I mentioned, it blends observed data with past weather forecasts. It includes things like wind speed, terrain height, ambient temperature, and relative humidity. And here, essentially, to create an ensemble of trajectories is what you're doing is you're moving one meteorological grid point in the horizontals here, the X and Y, and also 0.1 sigma units in the vertical each way. And the net result is you have a grid of 27 trajectories for one ensemble. So we would have 27 trajectories for a starting height above ground of 10 meters and 27 trajectories for a starting height above ground of 25 meters. So what the end result would be from running this model, and this is just an example for the 25-meter ensemble, is that you end up with these 27 unique trajectories. And what you can do is sort of try to grid those and show essentially a trajectory density. So on the bottom here would be the 27 individual trajectories. And then here we can sort of, if we grid them into boxes, end up with a sort of density of trajectories. So we can see that in this specific example, there are some trajectories that are coming from this area, but most of them are coming from this higher up area. So we have more confidence in, that the back trajectory is in fact here. So how do we link this to quantifying the likelihood of a cannabis cultivation facility impact? So here, sort of visualizing creating a half sphere around an individual CCF. And then if we have these back trajectories, we would want to see if we might find, if the back trajectory intersects with the CCF, then we might be able to say that the VOCs that were observed, or the odors that were observed in the odor report, would be potentially related to this cannabis cultivation facility. So it's a fairly simplistic type analysis. You can do this either for the individual facility scale or for saying from any facility. So I'll start with an individual facility. What you're going to do is for each of those trajectories, you're going to compute the minimum distance between each trajectory and the CCF. Right now, we're only doing this in the horizontal, and future work would be horizontal and vertical, but right now we're just doing the horizontal. And there's a buffer metric that gets calculated. So if the distance between the back trajectory and the CCF is more than 2 kilometers, the buffer metric is just by default set to 0. And then otherwise, it's computed in this way of 1 minus 0.5 times the distance divided by n, where n is the number of trajectories that did not hit the ground. So if a trajectory hits the ground, it gets eliminated from further analysis. So to visualize this, if we have this solid gold line is our back trajectory, what this example would be showing is a buffer metric of 1. So the odor report sort of perfectly cross-sects, has a cross-section with the CCF. And here this is what it would look like, obviously, if this is our half sphere of our CCF, and then it's not intersecting with that half sphere at all. This is going to be a buffer metric of 0. And this will give you a sense of a visualization of a buffer metric of about 0.5. So that would be done for an individual facility. We can also do this. It's that individual facility is a little bit conservative because, it seems conservative to say we want to know if this odor report came from this specific facility. But realistically, we likely might be more interested in did it come from any CCF facility? A number of these are quite close together as well. So what here we're doing is a very similar version of this, except we're computing the minimum distance between each trajectory and any CCF. So it could be that some back trajectories are closer to one CCF versus another, and this approach doesn't care. It just says the closest CCF is going to be the one we're going to be fitting to. We're going to calculate the maximum buffer metric in a similar way, and then we're going to sum all trajectories and divide by the number of valid trajectories. Okay, so a very similar concept is just saying instead of restricting the distance to a specific facility, we're going to say the distance to the closest facility. in this case. And the result of this is sort of, we ask the question of, which odor reports are likely due to CCFs. In this case, just to show you a visualization of what this looks like, this specific odor report, which is marked with an X here, has a composite buffer score of 30%. So what that means is you can think of this roughly like 30% of the trajectories are intersecting with these half spheres of these CCFs. So we've been able to look at 261 reports so far that mention cannabis or skunky smells. And right now, so far, we've been able to associate some back trajectories with 23% of them with a CCF facility. So this work is still in progress, but the goal would be, as the output of this is sort of developing a qualitative likelihood scale about how certain we are a specific odor report is going to be associated with a CCF, and looking at sort of percentage of different ensembles that are intersecting with these CCFs. So this is something we're working on to try to provide a sort of likelihood scale for people if they have these odor reports. So one way of analyzing them, but this is work in progress still. Okay, so that's some of the back trajectory work and what we can do with some odor reports. And now I want to sort of zoom back out and talk about just a critical review of the cannabis cultivation emissions in general, so that we can end with some fronts of action for people who want to get more involved in this subject matter. So this is work that was led by a PhD student in my group, Davi, who was very enterprising. I just need to sort of commend him. He is from Brazil, and he started his PhD in January of last year. He was unable to travel to Vancouver until July, and he really made lemonade out of the lemons he was given, because he said, you know what, I can't start, setting up any of the GC that we're supposed to be deploying. So why don't I try to do a critical review? And he submitted the proposal to do the critical review and wrote the whole thing and had it submitted by September of 2021. So I just want to commend him for working so hard under very difficult circumstances. So what I'm going to be showing you are some highlights of this critical review for emissions from cannabis cultivation facilities. So he assessed data from December 2020 to August 2021. So he looked at published studies spanning peer-reviewed publications to the gray literature. And the critical review was just accepted about a week and a half ago. And so I'm not sure when it's going to be available online, but it should be fairly soon. If you would like to read the critical review, if you're thinking about getting more involved in the cannabis cultivation space, send me an email or keep your eyes peeled for it, because it should be online very shortly. So some highlights of things that we looked at in the critical review, or obviously we were looking at this sort of VOC profile from cannabis cultivation facilities. We were looking specifically, we're going to do this monitoring and measurements of BVOCs. And there's this sort of question of what is the key tracer of cannabis cultivation facilities? So we're looking at other studies that have been done out there. And, there was a fairly I should say, an early study on cannabis cultivation, and early as in it came out in 2020, that was suggesting that, beta-myrcene appeared to be a good tracer of CCF emissions. This is a conclusion from outdoor measurements. So we can sort of see as samples are taken approaching the CCF and moving away from the CCF, the beta-myrcene here is in purple, and we can see the relative contribution of beta-myrcene is increasing when you're close to the CCF, and then it drops off again when you're downwind of the CCF. So we're like, okay, that seems promising, beta-myrcene. But things get a lot murkier when you're comparing different things. So this is a complicated figure, but bear with me. I don't expect you to sort of look at it in tons of detail. The first thing I want to point out here is component A of this graph panel, which is comparing 4 different plant varieties and the relative contribution of BVOCs. So we can see in some of these strains, and this is like elephant purple, has a huge contribution from beta-myrcene, from headspace samples above the plants. But in this first one, which is critical mass, very small contribution of beta-myrcene. So we can see it's not consistent across the different plant strains. Here in graph column B, they're showing 6 different ambient samples taken around cannabis cultivation facilities compared with background air. And again, we can see the contribution from beta-myrcene is fairly inconsistent. And then here are some samples actually taken inside some indoor grow rooms. And again, in one indoor grow room, or two of the four, there's lots of beta-myrcene, and in some others, it's barely there at all. So what we're learning is that it's not necessarily the most reliable tracer, and ideally, if you're going to be looking at CCFs, you're going to want to be looking at a portfolio of these BVOCs because it gets quite complicated. And so in terms of some reasons that we identified for potentially causing some of this variability, this is another paper that was looked at in the critical review, which is a heat map of BVOC and cannabis cultivars. The sample was of 240 cultivars in fluorescence, where they did genomic characterization of the terpenes. And the key thing to note here is this is sort of the different cultivars that were done here is that beta-myrcene is dominant in most samples, but in some others, it's practically non-existent. So this can give us a sense of, what mix of different terpenes we might be looking for. For example, terpinolene is less than 0.1% in 86% of samples, but it is the dominant VOC in the others. So hopefully this can give us some hint of the different BVOCs we should be prioritizing. But it seems this strain can cause this to vary a lot. And even just building up an emissions database or inventory of different strains and the BVOC emissions would be super helpful, because I've talked to a number of researchers in the US who are just sort of, doing this, like the study, this one where they were looking at the different plant strains. I mean, they did this in a garage in Colorado because it's very difficult to get funding to do the work in the US, non-existent at the federal scale. So there's just not that many measurements of different plant strains out there. So a huge gap in terms of understanding the BVOC variability. The good news is that there do seem to be some correlations between cannabis terpenes. So we can see here just some examples. The D-limonene is highly correlated to camphene, beta-myrcene correlated to beta-pinene, and terpinolene highly correlated to A-terpinene, for example. So we can hopefully leverage, we don't need to measure everything all the time. Hopefully we can also keep looking for correlations between BVOCs so we can target specific compounds and have increasing confidence that it is, if we're doing ambient samples, for example, coming from that CCF. The gold standard would be obviously to take the measurement in the cannabis cultivation facility and then outside the cannabis cultivation facility. We're working on some of those relationships, but it can be very difficult. It can be kind of a secretive industry to get inside and outside. Let's talk about this in terms of compared, like what this actually means in terms of a regional impact. So as part of the critical review, we also compared cannabis emissions to other crops. So if we compare it to other crops, cannabis plants do have higher BVOC emissions compared to a number of other common agricultural crops. So we are seeing in some cases in BC, if a facility that was a greenhouse for growing lettuce, for example, is now growing cannabis, we would expect to see an increased VOC loading from that. However, because the industry is still quite small relative to other sources, it's not like all of a sudden the total amount of VOCs in the region is going to be like hugely increased. It's a fairly small industry. So here we can see in Metro Vancouver, cannabis cultivation is fairly small. And the other data we were able to compile was from Denver, Colorado, where you can see, again, it is fairly small relative to some other sectors. If we put this in a different context, I found this actually like one of my favorite analyses that Davy had done in the review, is he looked at the odor emission units. It's sort of like an emission factor for odor. You can think of it that way. And he compiled some data from different cannabis cultivation facilities. And we think in the most extreme case here, a CCF that has 1,700 plants is about 800 odor emission units per second. Also, 800 odor units per second is about 1,600 chickens. So if 1,600 chickens, roughly equivalent to 1,700 plants, it's helpful to think, maybe one plant, one chicken can be a helpful analysis. I mean, I don't know how helpful it is, but I think it's fun. One plant, one chicken. Or perhaps in a more useful context, some of the largest facilities were estimated from Davi's work to be about half of a wastewater treatment plant. So this is an example from a wastewater treatment plant in Turkey. Again, not direct comparison to what might be happening in Vancouver, but this is the daily impact on odor of a 7,000 liter per second wastewater treatment plant. If we look at this by equivalence, 1,700 plants per CCF was about 60 liters per second. If we have 200,000 plants, that would be about 7,000 liters per second. And one of the larger CCFs in the Delta region has about 110,000 plants. So it'd be roughly the odor equivalent of half of the, this is that facility, half of that wastewater treatment plant. Another just way of thinking about the potential odor impacts. In terms of the literature on odor annoyance, an odor profile from cannabis emissions, it really varies person to person. So these are ratings of people who that they associate with cannabis. So here, you know, the strongest rating associated with cannabis was pungent, but you can see that some of these are like, oh, it's sweet and flowery, and, you know, skunky actually comes down fairly low. So in this paper, it was like, you know, these are actually kind of positive associations with cannabis odor, but it was noted from a review that this is kind of a biased sample. Most of the population that was polled to do this consumes cannabis recreationally. So it's important to keep in mind. Look at specific trained panelists performing olfactometry analysis. There's only 6 of them, but you know, they do have that sort of training. The cannabis emissions were found to be very low acceptability, especially at the end of the flowering stage, and of sort of medium to medium high intensity. So from the trained panelist perspective, acceptability is low, and that might be sort of related to why we're seeing more odor complaints in the Metro Vancouver region. The good news is that there are efficient removal technologies available, but adoption varies. So in terms of the review, we've also looked at different costs and technologies available, looked at by efficiency. So UV lights are the most efficient, but they're also the most expensive. From conversations with like WorkSafe BC, I've also learned that some people are like using ozone generators to react to the odorous compounds, but from those conversations with WorkSafe BC, they're sort of saying that, you know, companies are maybe not effectively containing the ozone, which is potentially, you know, making that problem worse. We're thinking about VOCs and ozone, and from an occupational perspective, not great. Some facilities use odor neutralizers, which I think of as sort of like Febreze for cannabis cultivation, which might actually be introducing more VOCs. So it's really variable what facilities are doing. I think there's a lot of goodwill in facilities to try to do things that are going to make sense. But yeah, we're hoping to actually do some sampling in some facilities later this year through a partnership with WorkSafe BC to better understand what people are actually doing and what the impacts on VOCs actually are. In the review, we also identified some key fronts of action. So we divided these out by different sectors, and there's specific details, or I think it was 16 that were identified in the paper for all these exciting researchers to work on. We divided them out also into interest groups. So for interest to regulators, we think it's important to create a database of terpenes and cannabinoid emissions for different cannabis strains under different growing conditions and life stages. And we're going to, obviously, regulators are going to be needing to set guidelines or increasingly review guidelines for exposure to cannabis-related terpenes, their oxidation products, dust in the workplace and outdoors, and how those are influenced by control technologies. Of interest to industry, some fronts of action that were identified would be potentially things like better understanding ventilation in the facilities by looking at things like CFD models paired with atmospheric chemistry applied to indoor greenhouses, could provide information on temperature, airflow, terpenes, CO2, and the goal here being, you know, maybe we could have something that's a twofold benefit where we could optimize the growing environment for the plants so that the industry is happy, and we could also improve ventilation from a health and safety perspective. So I think there's lots of opportunities there, and actually the current environment that we're in the mid-pandemic space is that people are talking about ventilation in a way that they weren't a few years ago, and so there might be more appetite for something like this now than there was even a few years ago. This is also the second one. It's from a conversation with some people who are actually doing research in the cannabis genetics space, where I think there's also opportunities to pair terpene concentration assessments with genetic modification to simultaneously increase economic value and decrease BVOC emissions. So this is something that, people in the genetics of cannabis strains are already working on. Like, how can we tune the precise genetics of something to provide a very specific experience? They're like cannabis sommeliers. And the conversation, we were having is like, what if, from that cannabis cultivars map that I showed, there's some where the BVOC ratios are very different. Is that something we can tune where maybe you have a very specific, your preferred cannabis experience while at the same time the plants are off-gassing less BVOCs? I don't know the answer to that, but we think it's going to be kind of interesting. And they're already looking at the genetics anyway. In terms of odor assessment, inventories of non-BVOC odorous emissions, very hard to come by. So everything here is really just BVOCs that we found in the literature, but there's other compounds we think are quite important, especially sulfur-related compounds. Continuing to do these olfactometry analyses of emissions at different plant life stages, very little data out there. Other tools to help assess odor, so odor perception networks, electronic noses can help do this, and hopefully some forward modeling of odorous emissions to develop some maybe minimum distance guidelines for where we should actually be siting some of these facilities. In terms of probably many people on the call, atmospheric chemistry and models, actually getting some measurements, we're hoping to do some of this stuff of concentrations downwind of photooxidized terpene products and ozone, just understand maybe what even some of these reaction rates might be, or impacts might be. Laboratory investigations as well of those reaction rates with key oxidants, improving the chemical mechanisms used in those atmospheric or dispersion models or in chemical transport models. I think they typically use like a bulk VOC mechanism, and we might need more refined things for cannabis specifically. Modeling odor persistence, including decay of odorous compounds via reaction, and any validation studies of tracer compounds of CCF. So hopefully we should be able to do some of that as well with mobile monitoring. And lastly is understanding health impacts of odors indoors and outdoors. So maybe more epi studies on measured concentrations of compounds and their health effects under short-term and long-term exposure, and ongoing analysis of how communities develop symptoms in reaction to CCF odors. Lots of things there to think about. So in terms of on the horizon for our research group or where we go from here, we are starting, just switch this off again here. We are starting to model odor dispersion from facilities. So these are some of those same facilities in Delta where we are looking at doing this dispersion modeling of the extent of the odor over time under different meteorological conditions. And this can be used to then potentially look at the number of odor exceedances that might be expected from a given facility. So here the density or the color density shows counts of 1 hour average concentration exceedances for odor. So we could have some projections of what those might be. This is all assuming a fixed emission factor for these plants. We hopefully can refine those over time. Also on the horizon, I'm very excited to do monitoring, which would be great. So I have a mobile van that we've been building for some time. It's almost done. It's done on the inside mostly. And it's technically called the plume van, but for this project, we're calling it the literal smell van. And we're going to use it to measure air pollutants, so ultrafine particle count, size distribution, ozone, and BVOCs around areas of interest in 2022. Later in 2022, we've purchased, and we're just waiting for it to arrive, an Aerodyne Vocus PTR TOF MS that can go into the van for real-time VOC characterization. And these results are going to be used as model inputs for chemical transport models. I don't think I added it, but we also, like I said, we're just finalizing this partnership with WorkSafe BC, hopefully as well, to actually take some of these same instruments inside a few facilities to help get some indoor-outdoor stuff. But it's going to depend on how many facilities sign up. I'm not sure how many that will be. Ongoing work as well going forward is on knowledge translation. So as I had mentioned, we do have a dedicated knowledge translation scientist on our team who's going to help convert this knowledge into action or policy briefs. We had our first outreach article available online called Elucidating the Community Health Impacts of Odors Using Citizen Science Mobile Monitoring. And here, like things like, word clouds of things that people are reporting from their odors. So, and we have not done very much of this so far, but Angela's even helped us for getting people to use Smell Vancouver with like giving feedback on our social media posts and things like that have been very helpful. Okay, so some concluding remarks to leave a little bit of time for questions. Understanding emissions from the cannabis cultivation sector, hopefully I've conveyed that it is complicated and we have enormous knowledge gaps. So I would encourage others in the Canadian research community to get involved. There's a number of reasons to get involved. It's very exciting science. And I also think that the Canadian research community has an enormous opportunity to be world leaders in the development of cannabis emissions policy and mitigation strategies. Like really, if this is done well and thoughtfully, which I think, we have every bit of talent here to do that sort of work, Canada could be the leader in this in the world. And the last thing I want to mention, I think probably not, again, particularly surprising to the audience of this sort of a seminar series, but for me personally, interdisciplinary perspectives have been so rewarding on working with this team. I know firsthand it can be more work to incorporate these interdisciplinary perspectives from the beginning, but the knowledge generation has been extraordinary. We started this project in a pandemic, and I think the amount of analysis and work and thought that has gone into it, despite all of these things, has been, just so rewarding to me. So if any of you are new faculty members or are grad students or think about making the jump into, professor positions, don't shy away from doing this interdisciplinary stuff because it can be really fantastic. And with that, I will thank you for listening. I'll have a little bit of time for questions. If you want to email me, I think many of you know how to find me. I'm easy to find. And I just want to thank the funding agencies, primarily the New Frontiers and Research Fund, and also the, Canada Research Chairs, which, keeps me at UBC, which is great. All right, I'm all done.
Thank you for a wonderful seminar, Naomi. We'll open it up to questions. Oh yeah, applause. There we go.
I can't see the chat. Do you want me to stop my share or just leave it? I'm not sure if people don't want to see
why don't you stop the share for now, and if questions require it, we can bring it back up, and hopefully it'll come back up. So
okay.
I'll check out the chat, but we'll see. I see a question. Who's got the question? Taylor's got a question, I think. Why don't we start with that? Yeah. And I'll check. Okay.
Thanks for the presentation. It was really good. I was really interested in this idea of measuring odor, which instinctively sounds like a really nebulous concept. Like it's a personal observation. So I was wondering, like, if you could elaborate a bit more on what an odor unit is, and also how are you controlling, if you can control, for the sort of self-selection of who decides to report odors. I mean, my first instinct when I heard this is that the people who are against this kind of research are going to say, well, oh, these are just the people who don't like cannabis that are complaining. So you've just created a really nice map of where all the people who don't like cannabis live. So, I mean, what would be a counter argument to that, I guess?
Yeah, that's a fantastic point in terms of, we are looking at some of the demographics of the people who are reporting this. I will say, actually, there's someone who runs a network in Vancouver that's like called, I can't remember what it's called. It's like, they're all about like pleasant odors in the region. Like ours are very skewed negative, but they're like the lovely smellscape of Vancouver. I can't remember what they're called, but we've had some conversations. We were expecting that, we would see a lot of people complaining about, these odors because they hate cannabis. We haven't had a balance of people who find it like not unpleasant. So they do exist, and I think there is no way around it. But these are the sorts of people who are complaining from a regulatory perspective anyway, likely, and we're just trying to capture a little bit more of their perspectives on sort of, the actions they've taken, their health experience of these sorts of things, just to try to understand in a more holistic way, what the impacts are. In terms of an odor unit, this is sort of a pseudo quantitative type thing where it's more based on like scaling based on people who are trained, is my understanding. I'm certainly not an expert in, like, Davy did a whole master's degree on odor units, so I don't want to misrepresent him, and I could maybe follow up with specific details. But my understanding is it's sort of like pseudo-quantitative, where you have these trained panelists who are, using these little smell, like they look like little megaphones you like attach to your nose, and they're taking a deep sniff and they're giving it a rating. So I would call it, pseudo-quantitative, and it's really just based on the training that you've received to be able to be like a registered smell quantifier. Is the way we're that's typically done. But I can certainly follow up with you and connect you with Davi, who can speak far more elegantly about this than I can.
Amaraskin, you've got a question.
Hello, Professor Zimmerman. Thank you so much for the fantastic and informative talk. I have a question about the chemical transport model. Actually, I don't have quite background in such models. So what I interpreted from your slides was that because only diffusion and advection are the underlying mechanisms behind the model, it's purely physical. But if I'm wrong, and it does include some chemical components, can you explain more how do you account for chemical transformations that some of these chemicals that may be responsible for cannabis odor, how they are being represented in your modeling framework? Because it seems that they are prone to atmospheric oxidation and they may already reacted in the time scale of these displacements.
Thanks for that great question. Yeah, so for this looking at the screening method, it is just a physical approach. It is not a chemical transport model. So you're absolutely correct that there could be reactions where by the time the person smells the odor that it's maybe reacted away. That is a limitation of this initial screening tool, but actually running chemical transport models is future work for where we're going. But we want to actually base it on, ideally, we're going to use the sort of built-in sort of data that we have available to us in emission inventories to do the modeling. But we're also hoping to actually wait until we have some actual quantitative measurements, both indoors and outdoors of some facilities, to make sure our inputs into those chemical transport models are as accurate as possible. So it's something we are planning on doing, is not currently represented in the screening approach, but is sort of future work going forward. And that's led by Amanda Jang and her team, whose background is in some of this chemical transport modeling.
Thanks.
Yeah.
I see Jeff has a question.
Oh no, Jeff always has tough questions.
Hi, Naomi. Nice to see you.
Nice to see you.
Really interesting talk. I had to join late because I was in another meeting, so maybe you covered it. I was just curious about trying to predict the transport and then the meteorology. And of course, we probably expect the odors are more acute when it's stagnant and stable, which are the times when the types of models, trajectory models, or any extrapolating meteorological data from somewhere else to your location is very uncertain. Are there better ways you have to get meteorology into here if you want to use those tools?
That is a great question, and I don't know the answer to that question. I'd have to forward it to Amanda, who would know better than me. My understanding is that this modeling that was done assumes stable environmental conditions, but not stagnant.
Yeah, or at least light air.
Yeah.
Yeah.
But I think like what we had approached this as is like, let's see what we can do from a screening perspective while acknowledging all of the limitations of the screening approach, and then we can decide, how we'd like to proceed to make this more, how many can we capture using this sort of screening approach, and then where do we go from there?
And maybe you could, capture some of the regulators' interest in this industry while it's there, and say that these facilities should have something like Met towers at their sites, and then you can see where it's going.
Yeah, possibly. We have some regulators on the call, so I'll leave that up to them. We can only make recommendations, maybe.
Yeah, we need really good Met towers there and low cost sensors all over the place.
Yeah.
Yes. Arthur.
Okay. Hey, Naomi.
Hey.
I think this is a very interesting project. I'm glad you got it off the ground. That's great.
Well, sort of. I mean, the monitoring is not off the ground yet, but soon. It's sort of.
I remember
feel like
we talked about this 2 years ago, and yeah, like you were, yeah. So it's great to see it's coming along. The chemical profiles with all the different terpenes, it's interesting that you can link them to, like, you could do the genetic analysis and actually like tune the strain and see which terpenes get emitted. I understand that like using a single species as a tracer may be challenging, but have you looked into using like groups of compounds? Because these terpenes have different structures, and you even show there's some sesquiterpenes and some monoterpenes. Would that be useful, like ratios of different groups of compounds, or maybe there's a specific group of compound that is coming from these specific strains, for example?
Yeah, I think that's where it's gonna. Where it's gonna have to go, right? Because some strains are gonna emit quite different profiles, and hopefully we can, like, to my knowledge, the analysis is not there to try to say, okay, here's like almost doing like, oh my gosh, the name for it. When you're like clustering the data, saying, you have certain characteristic ratios of different things, and saying, now we have certain confidence that this is coming from cannabis. I think hopefully with some of our measurements and other people who are doing these sorts of measurements, we can come up with sort of, characteristic clusters of things that we can sort of label as cannabis for a source apportionment type perspective. I don't think we're there yet, but I think it's needed.
Cool, thanks.
Yeah.
Great talk.
Okay, we are at the hour, but I think we can squeeze in one more question. So Miriam, if you want to ask the final question.
Oh my gosh. Okay. Well, thank you very much for a really thought-provoking, interesting presentation. And I'm also a fan of you and of Amanda Zhang, of course. By the way, the taste testing reminds me, I mean, if there's skepticism about it, then there should be skepticism about wine tasting. So, hey, just saying, okay. It occurs to me that there's initiatives for several years in Europe under REACH to look at natural mixtures of chemicals in terms of risk assessment, where most chemical regulatory schemes are attuned to single chemicals. And yet there are products, let's say cedar oil or, a lemon mixture that have very complex characteristics, chemical composition, with approaches onto how to actually move forward with regulation. It's a comment, not a, well, if you have any thoughts on that. Thank you.
I mean, I know that that's something that is a passion of Amanda's that maybe she, started to learn from when she was with you, is about, it's cumulative impacts, right? And I think, and maybe not specifically with the specific compounds and the cumulative impacts, but I think that philosophy underpins a lot of this work, which is like, there are these cumulative things that are going on. We expect there's the ozone and the chemical reaction impacts, and then there's people's perceptions of it, and even their annoyance with it, and the people who are the political aspects. I mean, all of these things have a sort of cumulative impact on the industry, and we're trying to have that sort of cumulative impacts philosophy, if not, exact framework in mind. So, yeah.
Sorry, it's not so much cumulative impact, but rather how do you address complex mixtures?
Yeah.
When we're accustomed to looking for single hazardous compounds, how do you assess a complexity of mixtures, which is always the case with nutricils and biologically, like naturally occurring mixtures. Anyways, I'll leave it there. Thank you very much.
Food for thought.
Okay, to close this off, if you have follow-up questions, Naomi is easy to find at UBC, so just look her up and send an email. I'm inviting everyone to use up your time there, but I want to thank you again for a wonderful seminar. I want to thank you for coming back for this virtual visit. I think we will try, when possible, to come have you back in person sometime. And I want to wish you and all our colleagues from BC and Vancouver much better weather in 2022. It can only improve from here, so let's just hope. But
let me know if any of you want to come to Vancouver too. It's normally kind of nice in the summer.
We'll keep that in mind when things are more possible to travel. But thank you so much for a wonderful seminar. So.
No, thank you for having me. All right.
So next month, we're going to have Sandra Odenthal from Scotiabank, who is their Vice President of Sustainable Investment. And so if you're looking to see how financial institutions are trying to address their contributions to climate, join us next month when Sandra, who's also a graduate from U of T Chemical Engineering, will be given an interesting talk. I don't know if it'll be equally interesting, but we'll see. So thank you all for coming.
Thanks.
Bye.